The last time that I spoke to you, we were talking about association cortex and the neurobiology of language. So I just want to kind of finish up that lecture. Okay, and then when we last spoke, we had a video about the split brain patients. And one of the real important things about those split brain experiments is it really, put the nail in the coffin as to what is a lateralized function and what's not. And it's complicated, right? So we understand that language is left lateralized. And there's kind of a lot of, hooey about left brain and right brain. But there are genuine functions that are more left or more right. And here's sort of a little list of some of those functions that we can assign to the right hemisphere. So obviously, you can. Obviously, you can use your right hemisphere. In some ways, there's evidence that you can read and understand numbers and things like that. That you can write. Obviously, if you're a lefty, you're writing with your left hand, controlled by your right motor cortex. So some components of language have been come out there. But your right hemisphere is apparently not able to take over the speaking role that your left hemisphere takes on. There's evidence for right hemispheric dominance in terms of drawing and puzzles, so some support for the idea that your right brain is a little more artistic. And then obviously the left hemisphere is clearly dominant for language. Which is not to say that in some cases you can get patients that have some shifting of language, especially if they have brain malfunctions early on. You can get some examples of pretty much everything. But this is sort of a good general construct. But most of what you hear about left brain, right brain is just hooey, okay? I don't want you to leave this class unless you can tell your friends and family that, so-and-so's TED Talk on, some new nonsensical left brain, right brain thing is just not based in evidence for the most part. Okay. Okay, so we're going to talk a little bit about brain stimulation. We talked about brain stimulation in terms of motor function, and obviously there's a big motor component to speech and writing. So it's sort of no surprise that if you stimulate in the motor cortex that you can disrupt some aspects of speech. So if you, on the other hand, if you disrupt Broca's area, you can disrupt speech or get speak hesitation. Okay, so we're sort of sensitive to those sorts of disruptions. Let's see, in posterior parietal lobe, there's an area where if you stimulate and the temporal lobe, where you can get word confusion and things like that. So sort of minor effects. And there was a really good BBC video that maybe I had it on YouTube. Maybe it's out there on one of the BBC sites that looks at, sort of takes you into the surgical suite and watch people sort of stop speaking when their brains are stimulated, which is just really cool. So if you can find that somewhere else, if I find it somewhere else, maybe I'll post it because it's fun to watch. But if you do this in various individuals, I think one of the difficulties is that there's a huge amount of variability among different people. So if you stimulate in different sites, this is, I think this is from one example, that all of those different sites will disrupt language to some degree. So you have a patient with their crania open and their brain exposed, and you do stimulation, and you're asking them to, count to 10 or count to 100. Excuse me. That you can disrupt speech in all of these different sites. And obviously, some of those correspond clearly to motor cortex and Broca's area and Wernicke's area, but some of them don't. And so this is a sort of a summary of stimulation experiments like that, where each of these numbers is the number of patients that had some disruption of language. And so you can see that there's just an enormous amount of variability. Okay, and then finally, in terms of language, we're going to ask, sort of a question that we all like to know is, where does language come from? do we have animal models of language? Obviously, animals communicate in various degrees, and you can have a whole graduate seminar on what exactly constitutes language. We could get very philosophical about it, but it's clear that animals, especially non-human primates, are able to learn a certain degree of symbolic language. And so sort of the famous examples are Coco the gorilla learning to do sign language, and then chimpanzees that can use a certain amount of symbolic manipulation to communicate. And these are probably the few examples that have been pretty clearly validated. Sometimes, obviously, with animal training, especially with a trainer that's very, very close to the animals, you can get a lot of sort of subjective wishful thinking. But I think these have been pretty well validated in the literature as being sort of genuine symbolic manipulation. Okay, and then finally, sort of our last little favorite model for language is in the songbirds. So songbirds, unlike a lot of other birds that use sort of simple calls, songbirds use complex song structures in order to communicate. They have to learn it, and so they're an example of vocal learning, which is thought to be an animal correlate of human vocal learning. Okay, and so you have these zebra finches, and we kind of talked about this a little bit before during the plasticity lectures. So here's an example. These are a zebra finch, so the male sings to the female. In this species, the female doesn't really sing. And so this is an example of a spectrogram. We have frequency versus time. And so the male has this very, very stereotyped song that he sings either at the female or sort of in general. And so the male has to learn this from his parent, so dad to son transmission. And we talked a little bit in the plasticity lectures about critical periods, and so there's a period during which there's sensory acquisition. This is time from hatching. Okay, so these are juvenile animals. And then there's a period where they're basically babbling and trying to figure it out. And they have sensory feedback from either the parent or from their own song, so they're listening to what they're doing, which is very critical. And then eventually, they get it, and they sing the very same stereotype song over and over and over again, which is great if you're a researcher because you can really define the song. But if you disrupt this, you can get sort of very human-like disruptions of speech learning. So here's an example where you have, let's see, these are sort of different swamp sparrows who have various normal songs. And then the juvenile will kind of babble. They'll do like little bits of song and try to kind of figure it out, just like humans will babble. And if they're not exposed to language, their song ends up being not only not what the parent would have taught them, so it's not innate. It's also very diminished. So it's a very sort of a poor song. And so just like humans who have not been exposed to language, they have very, very poor production capabilities. Okay. There's been some, a lot of interest in what is the genetic basis for song. So FOXP2 is a gene that's been identified. It was first identified in families that have certain types of language deficits. And it's specifically a motor impairment for speech production, but also processing words according to grammar, trying to come up with complex sentence structure, and they really have an inability to form intelligible speech. This is a transcription factor, so the gene encodes for a transcription factor, and it's really, really conserved across animals, and I think this has caused a lot of excitement in neurobiology. You can find it highly, highly conserved across animals, humans and mice, and it turns out in birds as well. And so a couple of interesting things, if you knock it out in mice, you can disrupt vocalization, cause motor impairment, but also you can find this gene in songbirds. And a researcher named Eric Jarvis has kind of led the way. He's been sort of at the forefront of genomics and neuroethology in birds. So he's looked at numerous species of birds and has a particular interest in birds that use vocal communication. Okay, and he found that not only is FOXP2, yes, FOXP2 found in birds, it's found at particular times when birds are learning song, so it's implicated in neuroplasticity, and particularly in vocal learners, less so in non-vocal learning species. So it's kind of a really interesting story there. All right, so that kind of sums it up for language. So we know that language involved localized and specialized brain areas for both production and comprehension, left lateralized, and that the split brain patient paradigm has really kind of led to a lot of understanding of this. All right, so let's continue on to our next topic. Yes, I know we have class now. All right, so we're going to move on to memory. And it sort of picks up where we kind of left off with the plasticity and experience dependent plasticity. In that we're going to talk sort of about what do we mean by memory. Again, we could probably have a whole graduate seminar on this. But we're going to look specifically at the role of hippocampus in memory. We're going to look at a very, probably the most famous neuropsychology patient called patient HM. And then we're going to talk a little bit more generally about what is the hippocampus in, particularly in rodents, and their role in spatial localization, and how that kind of talk a little bit about how that relates or doesn't. And then we'll finish up with a little bit on working memory and memory and sleep, which I'm sure everyone can relate to. Okay, so we can kind of divvy up memory into these different categories. How many of you have seen videos of these before? If you raise your hand here, I can't see it, so just at least a hand above your head. All right, there we go, thank you. Okay, so this is sort of familiar from various psychology classes. So memory, the psychologist, and this is human memory, into sort of two broad categories that you would call declarative, something that you're aware of, and non-declarative, which encompasses kind of everything that you're not aware of. And by declarative, we really mean your perception, your conscious perception of having been awake somewhere in the past. It also is your sort of reference knowledge. What did you learn in 353? And being able to, put that on an exam. So you're using your declarative memory to do that. Okay, and your general history. Okay, so that seems kind of clear. The other kind of memory that often we don't sort of associate with memory are things that you have learned. So particular motor skills, like learning to swing a tennis racket. Associations between things that you're maybe not all that aware of. Priming is sort of a popular thing to study in psychology. So this is sort of your unconscious awareness of certain relational associations. And then things like puzzle solving skill. We probably could add video gaming here. So you may not be aware that you've gotten better, but you play a game and then you play it again, you're better at it, right? And this has something to do with remembering, consciously, but it has probably a lot more to do with the fact that you've just done it before. Okay, so those are sort of categories of memory, and people can kind of argue over what counts and what doesn't count in each category. We won't do that. But then we also are aware that we have different time frames for memories, right? So remember, if you think back to our plasticity lecture, we talked about plasticity that happens at sort of different levels. So you have sort of an initial form of plasticity that doesn't rely on transcription. And then the long-term forms of LTE rely on gene transcription and protein synthesis. And so you can divvy up memory in sort of the same way with sort of similar criteria. But, and this is sort of not my favorite figure because it's trying to summarize data across humans and Drosophila and rodent models and kind of trying to cram it all into one thing. So don't overthink this graph. But generally, we can divvy up memory into something. This is the y-axis here is amount. You can think of this as the amount of memory retention versus time. So we all have a short-term memory, okay, that happens over a very short period of time. I tell you 7 digits, how long can you remember them, right? So that's sort of a short-term memory. And then on the other end of the spectrum, we have long-term memory. You know, these are your deep childhood memories, you know, what happened last year, or you know, knowledge that you have from you know, years past. And then we sort of have this sort of fuzzy middle here. So the overall memory is something that's sort of represented by this black line. But it can be sort of dissected into these sub-areas. So there's a level called medium-term memory. There's kind of this idea that long-term memory is something that's quote-unquote consolidated. We'll talk a little bit about what that means. Versus medium-term memory that's not consolidated. And then there's kind of this middle memory here that I think mostly comes from the fly research that we're not going to touch on at all today. But it's labeled ARM, A-R-M, as anesthesia-resistant memory. So if you've ever gone under general anesthesia, okay, there's sort of the last thing you remember is usually sort of before the prep. You don't usually remember the operating room. We're talking about just before you went under. And so there's some memories that are wiped out by anesthesia. And so everything kind of before that is considered anesthesia-resistant memory. Okay, but if we're talking about rats, how are we going to try and investigate what do we mean by memory or learning in these animals? And so the sort of most fundamental task or paradigm that people have used in rodent research is this thing called the Morris water maze. So it was invented by a guy named Morris. And, oh, I have a video. And I'll show you a little video. But the basic idea is that you have a training area, so sort of this circular pool of water. And rats and mice don't like to swim. I mean, they'll swim, but they don't like it. And so they will try to not swim. And so what you do is you put a little platform in the water so that they can kind of climb up out of the water. And they know they'll be rescued if you take them from that spot. Okay. And so the task is for them to learn where that platform is, which they do naturally because they just don't want to be in the water. Okay, so early on, you know, you teach them to basically get up on the platform. And then you have to teach them where is the platform. So what you do is you add something to the water to make it opaque. So they can't see the platform. It's just a little bit below the surface. And so they have to swim around until they kind of feel it with their feet. And so they swim and swim. So you put them in someplace. That's this little X. And they swim around, swim around, swim around, and then they bump into it. Okay. And then you take them out, you dry them off, and you put them back in, maybe in a different spot. And they swim around, and they might remember where it is. You have some cues. It's not a completely, you know, uniform area. You have some sort of cues. You know, you're in a room. And so eventually they figure out what happens. So eventually you put them in, and they just swim straight to the platform, and they're very happy. Okay, and then to test how well they understand that the platform is in that spot, you take the platform away. I know, it's kind of mean. But you take it away, and they swim to the spot, and then you measure how long they spend in that spot. So you can kind of measure how strongly do they think that platform is in that spot. Okay, let's see if I can get the video. Okay, so here's the little mouse. He's swimming around. I don't think this is very long, just to kind of give you an idea. And he swims and he swims and he goes right by it. And he swims and he swims. There you go. Okay. And you know, the researchers, they don't let them flail for too long. You know, during the training phase, they say, it's over here, and they kind of teach them. So there's sort of a time limit. They don't let them drown. So it's not completely. All right, so what does this have to do with hippocampus? Well, it turns out that hippocampus is necessary for this task. So remember, the hippocampus is our site for LTP, and we made an argument for, you know, why do we think hippocampus is so important for learning memory? And this is one of the reasons, is that if you lesion the hippocampus in the rat or in the mouse, it can't learn the task. So here's a control animal. Initially, it swims around until it finds it, and then it learns and goes directly. If you lesion the hippocampus before you do this kind of training, you know, it does sort of a normal first trial, which is important, right? Because you have to know that it's not failing just because it's got a motor deficit, or it can't move its legs. So you have to sort of test for all of these things. But it has a very normal first trial, but it hasn't learned. After 10 trials, it's still swimming around randomly, trying to find the platform. Okay, and so if you do this task, a normal animal will have a learning curve that looks like this. So initially, it'll spend a certain amount of time looking for the platform. There's a very rapid learning in trials 2 through 6, and so on. If you bring them back the next day, they're not so good, and then they learn rapidly. So there's sort of a multiple day effect too. But if you lesion the hippocampus, the animals just can't learn. They take just as long to find the platform on later trials as they do initially. Okay, so in the sense, this is memory in the sense of the animal has to learn the location of the platform. And almost all of our tasks involved in this sort of rodent test of memory has a spatial component. Lots and lots of these memory tasks have a spatial component, but in particular this one does. And so it's very clear that there's a spatial memory that you really need the hippocampus for in animals. So what about humans? Okay, so you might expect a certain amount of similar spatial memory deficits, but humans, it turns out that we know, because you can ask them and observe their behavior, that the deficits are categorized more as a sort of declarative memory problem. And we know this from a particular patient called HM, and so we're going to talk a little bit about episodic memory loss, known as amnesia, and how that's different from non-declarative memory. Okay, so patient HM, as is protocol in medical journals, you never name a patient for privacy reasons. They're often given initials, but this is a patient that has since passed away, and so now we know who he was. So his name was Henry Mollison, and he had epileptic seizures as a boy, terrible seizures, and so they tried to rectify this with surgery, removal of brain tissue in his temporal lobe in his early 20s. And it had really kind of a terrible effect on him. So he ended up with what we call anterior grade amnesia. Okay. And so this is amnesia, where if you walk into a room and tell him your name and have a conversation with him and walk out, wait 5 minutes and walk back in, He has no idea who you are. So he is someone who can't form new memories. He had some degree of retrograde amnesia, which is amnesia of his past prior to the surgery. So he has some memories of when he was a boy and where he lived and things like that. But he had a severe deficit of just could not form new memories. And this was in his 20s. And he only passed away, I think, well into his 60s. So he lived for a very long time with this kind of memory deficit. And so there was a great opportunity to study someone with this kind of damage and try and tease apart exactly what kinds of memory deficits he had. And so one of the things that was done was giving him a battery of tests of different kinds of memory. And so here's an example of a task, sort of a drawing puzzle. And I'll show you some on the video. And so he was able to do this task. So the experimenter would come in and give him a sheet. And the task was to trace the shape, so in this case a little star, only using the mirror in front of him. So there was a screen, he couldn't see his own hand directly, he could only see a mirror image. Okay, and if you ever try this is kind of hard. This is difficult to do. You can think that you know what you're doing, but it's hard to do. But after a few trials, you get better at it. And if you come back the next day, you're better than you were the day before after a few trials as well. And so you can have sort of a normal ability to learn. And so he had a perfectly normal learning curve for this sort of task. And so this is really the beginning of the strong basis of saying that the hippocampus is involved in declarative memory, but not in what we call procedural memory. So here's sort of a timeline of what we mean. If you have a brain injury at this point of the hippocampus, you lose the ability to form new memories. You may lose some recent memories, but you may preserve older retrograde memories. Okay, so we can kind of say this branch of memory types was impaired, and this branch not so much. Although I have to say, maybe we should point to this little box right here, because he could speak and converse, and had a repertoire of reference information, but he didn't know. He might have known who George Washington was, but he didn't know who the current president was. So this is a very weird state of affairs, right? You can imagine not knowing what you did. I don't know. As you get older, you start to have these experiences a little bit. What was I doing 5 minutes ago? I can't remember. But it's a very weird state of affair. But there's one of my favorite films. How many of you have seen this? Okay, I love this film. I think neurobiologists like this film. The film critics, not so much, but neurobiologists love this film. Because it's sort of embedded in this sort of murder mystery, but where he had anterior grade amnesia. And so from moment to moment, he didn't know what happened just before. Anyway, it's really, I think it's fun.
Can I ask a question?
Yes.
Can you go back to the slide, like, before? So for him, I know that, you said that he has, like, he can't form new memories, but, like, on the third day, he's still able to get better?
On the third day, so this is his performance on the first day, and he learns really well. The second day, he's a little worse, and then gets better. And then by the third day, he's able to. But if you asked him, he'd say, "I've never done this before."
Okay.
He has no idea how he knows how to do this, but he's good at it.
Okay. And that's because it's in his, like, non-declarative memory, right? Possibly.
Possibly, yeah. I have not seen any good explanation of where exactly procedural memory is. There may be some corticals, cerebellar, lots of other sites, but you don't need your hippocampus in order to do this.
But why did they decide to, like, try to fix his problem by going into the temporal lobe? Why didn't they just, like, sever the
So the question is, why did they do the surgery in the temporal lobe? Because I think the best information at the time was that was where the epileptic seizures were starting. And very commonly they start in the temporal lobe.
Oh.
Even now, it's clear that that's where they start. And even now, they have to do resections of parts of the temporal lobe to try to get otherwise untreatable epilepsy under control. But I think that was just based on the best information they had at the time. And they probably made a larger lesion than they needed to, but they didn't know any better. And they didn't know that what kind of damage would result from this. So, this was some 50 years ago.
Okay.
Yeah.
So was he aware that he had the surgery at all?
Was he aware that he had the surgery at all? I think he was moderately aware that something was wrong, but I'm not sure. I can't remember. I can't remember if he could state that. He would think not, because he would have lost the ability to form the memory that had happened. Yeah, but I think he was sort of aware that something was a little bit off. But he was generally, functional, happy, dressed himself, cooked for himself, could get around a familiar house, get lost in an unfamiliar house. Yeah. All right, so here's an MRI of HM's brain. So obviously people were interested as to, how extensive was the damage. I don't want you to, there's not that much to get out of here except that there was certainly, pretty extensive damage in the hippocampal region and probably parts of the parahippocampal region, entorhinal cortex, those sorts of things. And then when he passed away, he donated his brain to science. So his brain was fixed, flown to San Diego, and there was a live webcam to watch the sectioning of HM's brain. This was in 2009. And I have to say, I sat at my computer and I watched this for a little while. As boring as watching brain sectioning is, this was kind of exciting. So they sectioned his brain, and as far as I know, they're still trying to decide what. Actually, I don't know what came out of it, but they have a clear set of tissue samples from him to verify exactly what was damaged. All right. Anyway, neurobiology lore there. Okay, so we know the hippocampus is really crucial, but the hippocampus is kind of a hub, so it really talks to many other areas of the brain, including various areas of the forebrain and temporal lobe, and this part of the cortex that we'll talk about next time. But I'm going to show you a little bit of the video. So the person who really took on HM was this researcher called Brenda Milner, and she really kind of redefined the field of memory through this patient. So she kind of devised a number of these tests.
Allow me now to introduce you to a true pioneer. This woman earned her first degree from Cambridge in the 1930s. By 1944, she moved to Montreal, expecting to stay only a year, but she never left. With immense determination and, as she puts it, intense curiosity, she has spent her career investigating the mysteries of the human brain. Working with the legendary Dr. Wilder Penfield, she challenged conventional wisdom and wound up creating an entirely new field, neuropsychology. Meet Dr. Brenda Milner, considered by many to be the world's foremost expert on memory.
Brenda Milner was a Cambridge graduate when she and her husband came to Montreal from England in 1944. Milner initially taught psychology at the University of Montreal, but in 1950, she was enticed to McGill's psychology department by the arrival of Donald Hebb. Hebb secured for Milner a research opportunity at the Montreal Neurological Institute, working with the legendary Dr. Wilder Penfield.
that was June 1950. And of course, as soon as I set foot here, and I got so excited by it that I knew this is what I wanted to do.
Milner's job would be to study and test Penfield's patients before and after surgery that removed part of one temporal lobe, a new operation done to alleviate epileptic seizures. Dr. William Findell later became a member of that surgical team.
The preoperative testing, which she did, if she found the patient already had a memory deficit, we had to be much more careful with our removal at surgery because of that. So it's a very important and valuable collaboration in that sense.
I had become very, very interested. I was very well read about animal work and so on, and I had become very interested in the temporal lobes of the brain and you know, how they might work on the basis of studies in monkeys. And so I had definite ideas I wanted to try out. I'm going to show you a group of faces, photographs of faces, and I want you to study them carefully so that you can remember them. So therefore, I thought I will look for some, this is my PhD, I will look for some visual perceptual deficits, and I will have a hypothesis that I might see this more strongly from the right side than from the left. Hebb didn't really, but he thought I was being a bit daring suggesting that.
By 1952, Milner had accumulated enough evidence to demonstrate mild deficits on certain perceptual tasks, mainly after surgery on the right temporal lobe, less so on the left. Her hunch was right.
And, you know, I had to fight to get this accepted because they were subtle and because nobody was really had this faith in the right hemisphere. I had this interest in the right hemisphere.
But what Milner did next would change the course of her career.
You know, patients would come to me, and especially the ones with left-sided lesions, and they would say, oh, I have such trouble with my memory. And I wasn't there to study memory, you know.
But she listened and observed.
If a patient comes to you and says, I have trouble with my memory, you should not reply, and I didn't. You shouldn't say, don't talk to me about memory. I'm here to study perception, or I'm here to study the motor system. Don't want to know about memory. You should be very quiet as a mouse, and you should listen, and you should say what kind of memory. And well, if this is what the patient feels is wrong with them after this surgery or after this epilepsy, I should study memory, you know. And so from then on, because the patients said they had memory trouble, I switched the focus of my research to memory.
She got very hooked onto this idea of memory and developed her own tests and her own techniques for working out the details of these memory things.
Milner was concerned and challenged by the unexpected outcome in two patients who had developed severe memory impairment after what had become a standard procedure for temporal lobe epilepsy.
So Hebb had supported me for 1 year on his own funds, and I'd given up my tenured position at the U of M. Hebb thought I was really foolish. And I didn't know what I was going to do at the end of the 1 year. But then when we ran into this memory problem, Penfield came to me and said, we need you. You have to come. And he found me a little office at the neuro and so on. But neither Hebb nor Penfield gave me a penny for anything. If I wanted to do a test, I had to photocopy something. And I really had no money.
Milner pursued her methodical testing of Penfield's patients, and in 1955, Penfield and Milner presented their findings on post-surgical memory loss to the American Neurological Association. It caught the attention of a Connecticut neurosurgeon whose patient, known as HM, had suffered a profound and permanent loss of recent memory after a surgical procedure. Milner was asked to study HM, which she did over a period of several years, though HM never remembered her from one visit to the next.
I think one of the characteristics of Dr. Milner is her methodical, persistent, and very well organized testing of these patients.
It was one such test, the mirror drawing test, that revealed to Dr. Milner evidence that the human brain had more than one memory system.
For all of us, you or me today, whenever you get to the point of the star where you have to change direction, your hand does this kind of thing, and then, but not by doing, not by telling yourself what you ought to be doing, but just by doing it some way, by practice, you gradually improve and you keep a nice smooth line, and at the end it's beautiful. That is what you and I would do. But it's also what HM did. HM, over the 3 days that I tested him, and I had a lovely learning curve for this, and he got better and better, and he was doing beautifully at the end. And he had absolutely no recollection of the 3 days training he'd done on this. It was, that one trial was unique to him, experience, and he was proud of himself that he had done it so well. No recollection at all of all that had gone before. And it was this dissociation, you know, between his total lack of, between what he was aware of and what he had achieved. That was to me very exciting. You know, anybody asked me, you know, the really exciting moment, it was actually then, you know, looking with my eyes very wide at this going on. And this was early evidence that there's more than one learning and memory system in the brain, you know.
Milner's insightful analysis of HM's memory deficit is known today to just about every first-year psychology student, notes David Coleman, director of the MNI.
She's been able to take essentially a single patient, this fellow HM, and by asking the right questions, tease out the fundamentals of memory in a way that no one did previously.
Milner's discoveries in the 1950s began attracting new students and postdoctoral fellows, and at long last, some research funding.
Here you have a pack of cards, and there are 4 key cards in front of you. And what I want you to do...
In the 1960s, Milner turned her attention to the frontal lobes of the
where
brain.
you think you should go, and I will tell you whether...
She used the Wisconsin card sorting task to reveal a particular deficit among patients with frontal lobe lesions.
Okay. No, you can't change it.
Oh, I can't change it.
You just learn from it.
Okay.
What the frontal patients were doing was going on to pretend doing this thing which was incorrect. Even though they were being told wrong, they were not shifting.
This test revealed that damage to the frontal lobes affects a person's ability to adapt to change, more so from damage to the left frontal lobe than the right.
This is the work that I had the most difficulty getting published, because the whole climate of the time was to play down the frontal lobes. This is very hard to imagine today, but it was in the 60s, 1964, and I had more trouble getting this work published than anything, yet it has been very influential. So I'm quite proud of that, actually.
The thread running through Brenda Milner's research has been the complementarity of the left and right hemispheres of the brain. When she started her research career, conventional wisdom said that the right hemisphere was a bit of a spare tire. That didn't ring true with Milner, and she pursued a methodical testing of patients to reveal the unique functions of the right hemisphere.
Standing up in New York at Eastern Psychology meetings with these wolves, you know, this is where, you know, this was I didn't have to fight Dr. Penfield about being a woman or anything. I never had to fight because I was a woman, but the fact that I was daring to claim something for the right hemisphere in New York with this crowd of you know, self-confident young psychologists and their professors was tough. but I believed it, you know, I believed my observations. You know?
So Brenda is an extremely clear thinker, and she's very persuasive. When she's convinced of something, she can argue very effectively on behalf of whatever she's convinced of. And she's also an extremely keen observer. So you break that combination, and you have a formidable scientist.
Milner's many research breakthroughs were so groundbreaking that they led to the establishment of an entirely new discipline within the field of psychology.
She's often given credit for establishing what we call clinical neuropsychology, that is, psychology directed toward patient care and understanding patient's behavior, as being one of the individuals who have been largely responsible for developing that field. And if you look at many hospitals all over the world, the individuals who are doing patient testing and examination of patients from a psychological point of view have been trained here at the Neurological Institute by Brenda and her successive team who followed her.
Her contributions to memory probably would not have been done by anybody else. And I think that's her uniqueness. She's recognized as the world's authority on memory.
At 90 years of age, Brenda Milner is still actively pursuing her research and passionately sharing her experience with students and faculty.
So I suppose more than any single finding, I look around our group, our cognitive neuroscience group is really big, and I look around the people around the world who train here with me and with my younger colleagues, and I think I'm proudest of that, you know.
If it weren't for Brenda Milner, using this unique model that she recognized was unique, we would know much less about memory, the psychology of memory than we do today.
All right.
That's great.
I think we can agree she's awesome.
She's not gonna use it.
I believe so.
I know.
Is she still alive? I think so. Yeah.
It's so crazy.
All right.
Is she still working?
So she's still working. In fact, we had one of her recent trainees, who's now a professor in Canada of her own, who came and talked about some neuropsychology research recently. So she's still training.
That is so amazing.
Okay, and there's another video here where you can actually sort of meet someone who has anterior brain amnesia. I won't play it here, but you can go ahead and take the time to read it, to watch it. It's really interesting. Okay, but we need to move on to sort of another topic in the hippocampus. And as far as I can tell, this is not covered in your textbook, which is just crazy. And I think because it sort of doesn't sort of fit in the general theme of the book of what the hippocampus does, is memory. But throughout the 70s to 80s to 90s, it's really been a dominant paradigm of spatial localization in the hippocampus. And so we're going to talk about that. So obviously the damage to the hippocampus impairs spatial memory in rodents. There's evidence from non-human primates and humans, obviously, that we have non-spatial memory. But in rodents, that's kind of what we can test. And so, back in the 70s, people started recording from the hippocampus in awake behaving animals doing these various tasks. I think under the idea that we would understand learning and memory. And what they found, more than anything else, was this thing called place cells. Okay, so they found that the pyramidal neurons in the hippocampus responded to exactly where the animal is in the Morris water maze arena. And when they started doing mazes, they found that these neurons would respond to being in a particular spot in a four-arm maze or an eight-arm radial maze and so on. And so this is really, this was a incredible finding that's been followed up in numerous other studies. It has now sort of become a field of itself, looking at these place cells. And so these are space-specific firing in response to position. And then following up on that is a more recent finding from the early 2000s, not even the 90s, it's like early 2000, 2005, I think, is this finding of what we call grid cells. So these are cells in the entorhinal cortex, which is tightly coupled to hippocampus, that are also position or space-specific fields. But as I'll show you, they have this incredible, completely unanticipated finding that they have responses that form basically a hexagonal grid of space. And we'll look at those in a minute. And in 2014, 3 of the key people in this field were awarded the Nobel Prize. Okay, so the paradigm is to record from the hippocampus. So you have an animal that's exploring, you record their position, you record the activity in the hippocampus, and you get something that looks like that up in the corner there. And I'll show you a sort of expanded version. And this is called a place cell field. So in this kind of paradigm, so the square is the space, the field that the animal is in. Okay. The gray lines are tracking the animal as it explores the field. And the red dots are the position the animal was in when a spike was fired. So it's sort of a positional raster there. And so what you find is that if you're recording from a particular individual neuron, that it only fires when the animal is in a particular position in space. And then you'll record from another cell which has another position in space. And so you could tile the whole field with this population of neurons that respond to different parts of space. A couple of important characteristics of place cell fields are that they're sensitive to cues. So if you put the animal in an arena with, a poster on one side and, a star on the other, and then you shift it around, the place fields will shift. So if the place field was originally here, and you have two sets of cues, and then you rotate them 90 degrees, the place field rotates 90 degrees as well. The same cell can respond in different contexts. So this is an example of if you put the animal in a square field versus an animal in a round field, that one particular cell might respond to this position in the square field and a different position in the round field, and so on. So there's a very deep literature on that you can go look at if you want to know. I'm not going to summarize all of it here because I don't know all of it, but it's very, very interesting stuff. Okay, so let's look at an example of a place cell field. So in this example, what you're going to see is you're going to see an animal on a track. Okay, so this is just an animal running on a linear track around the room. And you're going to hear spiking. So this is a multi-electrode recording. And so you're getting spikes from a number of different individual cells that are color-coded on this plot. And they're going to be plotted on here and on here. So watch the colors. And so people can do this by sorting this experimental procedure called spike sorting that we won't get into. But in the end, what you find is that these different neurons will respond to different positions. So let's take a look at that. It's very short. So you start to get the idea. All right, so this animal had blue cell firing when it was in this portion of the track and orange firing at maybe two positions. And the bigger the track, the more complicated the fields can be. So what I showed you before was very simple. Get the idea. Okay, so these are hippocampal place cells that only fire, and they also have a few other aspects. Okay, so generally the place cell characteristics are that they're restricted to a small number of, or sort of a small area within a particular field. They're sensitive to cues. These are not really learned. These are things that, the activity that happens the minute you put the animal in an arena. It may respond differently in different arenas, but it's always there. On the other hand, if you put the animal back into the same context day after day, and assuming you have a truly stable recording, it looks like these responses are pretty stable from day to day. So they're both flexible and stable and responsive to multiple environments. And so they're thought to be really critical for spatial memory. In the entorhinal cortex, you see something really wild. And so this is an example of a grid cell. So people knew that there was a strong projection from entorhinal cortex to hippocampus. People were trying to understand how are these place cells formed. And when they started recording from entorhinal cortex and using relatively larger and larger fields, what they found is that the response field for a single neuron, so this is, again, you have the track of a rat and a single neuron, and the firing forms this sort of lattice, okay? So it's forming what's called a grid cell in the sense that it has this sort of triangular or hexagonal lattice formation, which really was amazing. The first time I saw this, I went to a conference and I hadn't really heard about it. I wasn't following the hippocampal field and I saw a talk. They showed this and my jaw just hit the floor. I mean, it was really that surprising. Nobody expected anything to look like this. Ergo, Nobel Prize. And one of the really interesting things here is that each cell has this sort of grid formation, and then you have multiple cells that may have the same grid but kind of sort of the same size grid but kind of shifted over. Okay, so you're tiling the space with these grids, and the spacing between those vertices has a certain size at one level of the entorhinal cortex, and then it gets enlarged at other sizes. So these sort of spatial fades, or the spatial resolution here, changes in different portions of the vertebrae. So it's really interesting. People have found this in other animals. I could have shown you similar cells in the bat entorhinal cortex. Okay, so this seems to be a strong phenomenon. People are still kind of trying to figure out, which comes first, the place cell or the grid cells. How do you get both of these things? And it's a really interesting problem that nobody has solved yet. But so here's a sort of more elaborated version of the hippocampal circuit. Remember, we have dentate gyrus, CA3 to CA1. Well, over here you have entorhinal cortex. So it receives input from hippocampus. This is subiculum of the hippocampus. And it provides input to multiple regions in the hippocampus. So these are sort of reciprocally interconnected areas. And so it's kind of a really thorny problem. Okay, so in addition to place cells and grid cells, you have a few other cell types. You probably noticed that if the animal turned its head, sometimes you got additional firing. So there are a type of cell called head direction cells that don't depend on a particular place, but depend on the position of the head. And then there are boundary cells that kind of tell you I'm next to the wall, and so on. And so all of these together, the idea is that they create kind of a spatial reference. And it's really crucial for being a rodent who's running around looking for food. It has to remember, where did I see food before? And how do I get back there? And how do I know where I am? Which is true for all of us as well. Especially those of you, if you've gone looking for my office upstairs, you sort of circle around going, I don't know where I am. And then all of a sudden it clicks. Okay. I like to think that's your hippocampus kind of jumping into action there. So then animals that cache food, it's been shown that they need a hippocampus in order to perform this behavior. So certain animals like chickadees that put food, or maybe squirrels that put food in particular places, and they have to remember where those places are, requires hippocampus. All right, so obviously the hippocampus is not working alone. It's interconnected to various areas. And this is probably best understood in the working memory paradigm. I know we talked about this a little bit earlier. So working memory is kind of that short-term memory of, keep a series of numbers in your head, or it's required for that Wisconsin sort task. You have to remember what you did just before, whether it was, yes or no, in order to learn. So you really need working memory in order to learn anything. tightly correlated with that. Okay, and this is just a hippocampus is interconnected to lots of other things, so has strong connections to entorhinal cortex, parahippocampal, perirhinal, and so on. Through those connections, it connects to neocortex, and then through other various nuclei in the frontal cortex and prefrontal cortex. Okay. So this is sort of another variation on the delayed activity cells that we talked about before, where the animal has to do a delayed task. It has to keep in its head where the stimulus, where the food was, and then find it. And so here's another example of it's provided the stimulus, there's activity in the prefrontal cortex, there's continued activity throughout the delay period, and then during the response as well. Okay, so this is sort of the neural correlate of short-term working memory in prefrontal cortex. Okay, and then the last topic I want to bring up is the idea of sleep and memory. So you've probably all sort of read or heard that you need to sleep in order to learn. So there's a sort of body of research on this topic, and you have probably already come to your own conclusions that sleep deprivation is bad for learning, right? Yeah? Okay, so here's the evidence that sleep deprivation is bad for learning. So here's extent of memory encoding for whatever task it was, I don't remember what it was. So you have a certain amount of memory, and with sleep deprivation, there's definitely an impairment. Okay. We can show that sleep deprivation actually reduces hippocampal activity. So here's a normal person hippocampal activity. This is like an fMRI study. A sleep-deprived person has reduced hippocampal activity. That if you can elicit extra slow wave sleep in a sleeping person, that you can get enhanced memory. This was a study of using transcranial stimulation to elicit extra slow wave sleep. And the idea is that this can help. But in general, good sleep is good for you. There's some evidence that naps help too. So there was a series of studies where you do a learning task, and then you test for memory sometime later. And there's sort of a standard, you know, if you test them some hours later, this is like a syllable learning task, that you have sort of a standard loss of memory, loss of performance. But if you get a nap in, you do better. I think it's still kind of up in the air whether this is, you know, sleep actually helps you versus sleep kind of shuts you down and you don't have interfering experiences. But anyway, I'll take naps. I think that's a good idea. Okay, but it's really kind of an open question of what exactly is happening during sleep that might help you learn, right? So obviously you're not going over your notes in your head, really, or are you? Okay, so some of the evidence for what might be happening during sleep has come from recordings from animals that are sleeping. And some of the earliest evidence actually comes from birds that were sleeping. But this is sort of an example. It's been kind of taken over by the rodent field. So this happens a lot. Non-rodent people will show something, and then the rodent people will all jump on it and get famous for it. Okay, so the idea is that, so say you're recording from the hippocampus after a day where you didn't ask the animal to do anything in particular, there was no learning task, and you record from various neurons in the hippocampus, and you look at their activity levels, and you don't see really any sort of pattern or correlation. Okay, and the idea is that these have sort of intrinsic drive and some external drive from cortical inputs. And then during the next waking behavior, during the next day cycle, you're training the animal. Okay, and while you're training the animal, you're getting this patterned recording. So imagine that animal following that track, right? So if the animal follows the track and gets rewarded, and follows the track and gets rewarded, you have this sort of pattern of activity. Okay, and it has a particular timing to it because the animal is traveling in a particular path where first one cell is active and another cell is active. And if you think back to your sort of Hebbian plasticity, you can see that you're kind of getting a chain of activity. So if these are all initially sort of weakly connected to each other, if you then correlate the activity in a temporal way, one neuron after another, you could elicit plasticity that would strengthen the connections of that particular trajectory of activity. And so if you record, you can sort of order these neurons however you want, and so you have this temporal pattern of the activity. And so it turns out that if you then record from the hippocampus that night that the animal, it's asleep, but its brain will essentially be running through the same pattern of activity. And that was kind of amazing to people who first looked at that. And so it's like the animal is running the maze in its head. There are interesting temporal components to this. So when the animal is actually running in the track, it has a certain sort of timing relationship between those firings. During sleep, that time is compressed. Okay? Nobody knows why or what the effect of that is. And then the idea is, well, how does that help learning? And the idea is that you're basically strengthening those connections between the hippocampal neurons, but then you're feeding back to the cortical circuits that were responsible for that activity in the first place. And that you're basically consolidating, this is where we get into the consolidation argument, that you're consolidating the information from the hippocampus back into cortical circuits. Okay, so the idea is that you're kind of reactivating whatever was going on in the cortex and consolidating those associations. Okay, so memory consolidation may be one really critical function of sleep. And we probably do it, and lots of other animals probably do it. Sleep deprivation impairs your memory, so sleep before your final. Slow wave sleep. Does anybody have one of those like sleep monitor things? Can you no? Okay. Yeah, I know somebody. No, it's not me. I know somebody who has one of those. And like follows the peaks and valleys of their sleep. And the idea is that some sort of replay of your waking experience is causing reinforcement of this. So there's good evidence that you should sleep on what you're learning. All right, so we're going to stop there.